Let me tell you right at the outset that I never learned the philosophy or psychology of teaching and learning, and I never talk about it. Okay. So the only thing I'm going to talk about is my experience of doing something crazy with students. As a student, I was crazy. I was a bad student. I was a backbencher. I never paid attention to anything that was going on in the classroom. At some stage, I started asking myself, why is it that I'm not finding this interesting? Okay. Now, this was just when I started gaining some interest in science. And that was when I had completed my graduation in science. When I started feeling some interest in science, I started asking myself, why is it that I never found that classroom science interesting? And then I decided to become a teacher just to experiment on can we make it interesting. So after so many years of teaching, actually it's very unfortunate that only recently I perceive myself only recently that now I know a little bit of teaching and I have retired. Okay. So my journey of science was a fakiri, completely. Okay. Nothing in hand, no achievements, okay, no awards. Okay. So that is called fakiri. So I had no brilliant academic record. I didn't go for PhD after masters. I never did a postdoc any time. I had no research position until in 2009 when I shifted to ISER. Okay. I had a poor funding record. Even I have a poor funding record except for a brief period. And then I don't have any focal theme of research. People often ask me, what do you work on? And I said, nothing. I had very few PhD students, and maybe I had a few publications which were in the flagship journals, including Lancet, American Naturalist, and so on. But I always thought that that was a byproduct. Okay, that's not what I wanted to do. Okay, but it somehow happened. And it happened because of this upside-down story that I pursued. Okay. Generally, people perceive that if you want to do a research career, then you do your graduation first, you do your bachelor's, master's, then take admission for PhD in a good institute, and that is where you begin your research. Now, I tried to turn the story upside down, and I said that no, I want to do research first, and then we will start learning. Okay. Or we will use research as a tool in learning. Okay. And it is my own experience that your science learning begins when you start your research. Till then you might be preparing, but you are not really learning science. So I'm going to tell you the story of this upside down thing. Okay. So what I evolved through trial and error, after making mistakes for a long time, for several years, that the reason I was not satisfied with what I was learning in the class, and perhaps why I am not able to deliver why I wanted to deliver to my students, the biggest hurdle is the curriculum. Okay. So the first thing in science teaching, I thought that we must remove the curriculum from science. Okay. And then science becomes interesting. So this was in a way an attempt to do non-curricular science. Okay, now people have used the terms extracurricular and so on. I'm using the term non-curricular. You have no objectives. You have no goals. You don't have to achieve anything. You are not working for any degree or anything. Right? Maybe you are doing something because that interests you. So why not start with that? Then, most of my research is with undergraduates. Well, this is not new because today it's a culture that in their second year or third year or maybe first year of masters, people go for summer training to various institutes, labs, and they do something. Sometimes they come out publishing a paper or being a co-author in a paper and so on. But that was not what I intend to do. In our model of doing science, well, the difference is in those summer training programs or programs that the academies have floated and so on, there is a lab which is, or an institute that is allotted to you. You go there. The lab has a research program, right? And they give you some project in that. In fact, I find this very funny, this very word of give a project. How can a project be given? The project has to evolve. Okay, it has to come from the student's mind. It has to begin with a question that a student asks. Okay. You can't give a project. Now, even I kept on getting requests. Now I feel better because I have retired. I kept on getting requests for students for summer projects and give me a project. Okay. I said, that is absurd. You can't be given a project. Okay. You have to ask a question. It has to be your own question. And then let us see whether we can address the question. I'm not saying answer the question. That is too far off. Whether we can address the question, whether we can think of how it can be resolved. What experiments can we design, whether we do it or not is a further question. What experiments can we think of, can we design to do, and so on. So, essentially, it is centered around undergraduates, their thinking, their feelings as well. Okay, and okay, let me tell you how it starts and how it comes out. But then when I started doing this, the biggest advantage was there is a high risk tolerance, which is not there in PhD system. Okay. Now, PhD is a huge investment. Students spend 5 years, 6 years, 7 years of their 20s, of prime life. Imagine 6 years of prime life is a huge investment. At the end of this investment, you can't afford to say that, oh, I didn't get anything. Right? And therefore, a PhD has to be a safe game. You start working on something where you know that you are going to get results. You can't take a risk at PhD. Neither the supervisor nor the student can afford to take a risk at PhD. But undergrads can. They can ask a question for which they don't find an answer at the end. They are unable to progress in spite of intensive thinking and discussing. They are unable to progress. That doesn't matter because they are getting their degree anyway. They have nothing to lose. So there is a huge risk tolerance in this system. And high achievements will often come when you are ready to take high risk. Okay. And working with undergrads has a huge risk tolerance, and therefore, in my experience, undergraduate research often works better than PhD and postdoc research. Okay, because they can take risks. And then, I worked in a college like yours, and the biggest advantage of working in college is you have nothing in hand. Okay, no equipment, no funding, no labs, and that is a biggest advantage. And the reason I say it is the biggest advantage is because you have nothing in hand, you start from scratch. So the entire thinking is yours. Okay, you are not copying anyone. Okay. And the specific advantage of being in a resource-poor environment is this, that the frontiers of science are moving too fast. Right? And if you want to follow them with the kind of setup that you have, you will find that it is just impossible. By the time you learn what is being done on the cutting edge today, it's outdated. Okay, so you can't be a good follower. And if you can't be a good follower, then you are left with only one option, and that is you got to be the leader. Because you can't be a follower. Then the only option is you have to be a leader. And the way you can be a leader is ask an original question which no one has asked so far. Okay, so if you ask, and in science, not even 1% questions have been asked so far. Forget about answered. Okay, not even 1% questions of the entire set of possible questions, not even 1% has been asked so far. So if you come up with a your own question, original question, there is a high probability that no one has asked this question so far. And if you invest, if you investigate, if you invest your time and efforts in it, there is a chance that you can come up with something that is new to science. Okay, so this is the relationship between risk and success. Okay. If you want to be safe, you can be perfectly safe, but you won't be able to achieve anything. You won't be able to go ahead. Well, so because, as I said, there is a high risk tolerance with undergrads, we can take risk, and therefore we are completely uninhibited. Okay. Well, there are something called subjects that you have to choose. So because I worked in the microbiology department, my first audience was a group of microbiologists, but that changed very soon. Katta is a known concept. Okay, there have been so many kattas. What was new? Well, by the definition of katta itself, there is no formality. There is no subscription. You don't have to be formally a member or subscriber of anything. If you walk in, you are a member. The moment you walk out, you are out. Okay? So there is a complete walk in, walk out thing. And we come together for an hour or two. In fact, the starting time is 6 p.m. There is no ending time. Okay? But we go on. There is no pre-decided speaker. There is no agenda. There is no focus or intention. So the discussion goes on drifting randomly. On a given day, we might start with some basic question in physics, end up talking about human behavior. Okay? There is absolutely no innovation. And then, during this, a large number of ideas are churned out. And then, when you have a large number of ideas, further path is left to chance. This is something that I call natural selection. If the idea is interesting, someone feels like following it up. Okay? When you talk about 100 ideas, you find that about a dozen of them have been picked up, and someone says, I want to work on this. Okay? So then let us start thinking. Then there is no commitment, zero commitment. If you start working on something, it is not necessary that you should complete the project. Okay? If you feel bored, if you feel like giving up, you give up the moment you feel bored. Right? So then, this is natural selection. So ultimately, what comes out of natural selection, obviously what is more interesting will go on more steps following. Okay? And therefore, what you come out is often extremely interesting and extremely catchy. Okay? So, just a span of things that came out of this katta system. So we published things from microbiology, ecology, evolutionary biology, cell biology, biology of aging, sex and sexual selection. So we worked on a variety of things, diabetes and obesity. Some of them were started by undergraduates, and they were later on followed up by PhD students. Okay, a project started by first year or second year. Later on, there were 2 or 3 PhDs on the theme because the theme itself was new and expanded. Okay, then today, I'm not a person of medicine, okay, and my mother wanted me to be a doctor. Very fortunately, I didn't score well, and I didn't go to medicine. Okay, but today, in one major hospitals in Pune, a new clinic is established which is based on my concepts. Okay, the concepts were originated talking to undergrads. Okay, and that is in the area of diabetes, obesity, and okay. So all this span of things coming from a single lab, as far as I know, is unprecedented in today's world of science, because today a lab works on one protein for 25 years. Okay. This is just the diametrically opposite of that. We put our hands in everything. This is because students come with different interests, and as a teacher, I have to cater to them. So I have to put my hands in several subjects. I can't say that this is my subject and this is the only thing that I will do. If you want to cater for students, then you have to be so, versatile, dynamic, and so on. This is like if, if, if you have a restaurant and people come to you, then they would expect a variety of things and you have to cater to them. You can't say that, okay, vada pav is a different thing, okay, which is a different game. And you can say that we sell only vada pav, nothing else. But college is like a restaurant, not a vada pav market. Okay. So now, just to give you a glimpse of what are the different kinds of things that we did in the katta. So for example, Dictyostelium is a slime mold, and that develops, and that has a very interesting life cycle. But there is social life there, and there are several human-like elements in the social life of Dictyostelium. So for example, some of the cells cheat. They don't do the function that they are supposed to do, but they take advantage. Okay, so there is a phenomenon of cheating among microorganisms, right? And we found several parallels in human society and in the microbial society, right? And then we looked at how does the dynamics of cheating work in a microbial society. So this we published as a paper in 1997. That time teaching was a new concept. Today a lot many people have talked about cheating. In fact, cancer cells in a way are cheater cells. Right? They grow at the expense of the body physiology, but they don't do the function that they are supposed to do. Okay. So the concept of cheating has become quite common today, but at that time it was fairly new. Then this is something that we published in the Lancet, which came from the first year class. Then it was followed by a couple of master students. At the end we had a small Lancet paper. And what we found was, among the sample, which the first sample was the classroom. Then we did a survey of Pune city with a few thousand families and so on. It turned out that families that take more care of water suffer more from diabetic diseases, from diarrheic diseases. Okay, and this was published by the Lancet 1997. Then we worked on crazy ideas like theory of mind in birds. And I have no background of psychology. This idea came up from our study and exploration of birds around, and then later on this paper was, what you mentioned, was a paper in the curriculum of the Department of Psychology of an American university. Okay, so that was that. Then social life of mitochondria, okay, a crazy idea, but mitochondria do have a social life. Then agar to agarose by fermentation. This was an accidental finding, and a patent was filed on this, that a bacterial culture that we got accidentally was able to degrade agaropectin, leaving agarose behind. So you could start with agar and get agarose. The difference in price is something like 100-fold. Okay. So then we talked about genetic algorithm for musical creativity, human mating system and stability of marriages, game theory and stability of corruption in the society, and so on. All these are essentially ideas that have a mathematical, we found a mathematical basis to that. Very interestingly, something very parallel to what Rajendra said. I was a total maths failure, okay? And in what was called pre-degree that time, I thought that was my last salute to maths. I will never do maths ever again. Today, more than 50% of my work is in mathematical biology. Okay? I started learning maths when I discovered that there is very interesting maths in biology. And then I had to do all over again, and this time I did much better because there was no teacher. Okay. I had to do it entirely myself. So we do work substantially on mathematical biology, and an important thing, an important reason for that is, since I worked mostly in an undergrad lab, we could easily do maths. You don't need a lab, you don't need funding, you only need to think and write a few equations. right? So one of the reasons why I came to mathematical biology and computational biology is you don't need a lab. Okay. So that is doable, and particularly for our undergrad labs, computational biology is extremely valuable because you can do it. Your computers are not that costly. You can easily have them, and all of us and this new generation, all of them have laptops, and that is often sufficient to solve complex problems. Okay. So one of the concepts that one of the fields was evolution, because the question why in biology is extremely important. I had an excellent teacher when I was a student, and he was a very popular teacher. He made the subject very interesting to us, but he used to say one thing. He said that never ask the question why in biology. You can ask what, how, and so on, but not why. Okay. This is an old statement. Okay. This statement collapsed in 1859 when Darwin published his book on natural selection. Okay, because the question why in biology can be answered only in the light of evolution. Okay, why we are what we are? Because we evolved this way, and the reason we evolved this way were because of specific selective pressures under those kinds of ecology, those kinds of environments, and so on. So the question why in biology is philosophically very deep. And that depth of philosophy is something I experienced, undergraduate can easily grasp. So evolution very rapidly became the central theme. And then there are several themes that were extended, that got beyond the katta, and which I am pursuing now as my lifetime mission. Okay, so one of them is tracing the evolutionary origins of type 2 diabetes, and then it turns out to be behavior-centered. So why, the reason why increasing number of people are becoming type 2 diabetic today does not lie in the diet, it lies in the behavior of people. And there is now substantial neurobehavioral data which is linked to the endocrine and metabolic systems and so on. Okay. So, so then, bacterial plasmids is something that is very wonderful because there are very few plasmids in a cell. Now, plasmid being small can replicate very fast, right? And therefore, if they use the same machinery, but the plasmid replicates 100 times faster than the chromosome, the cell will be full of plasmids. But no, plasmid replication is regulated. Who replicates plasmid replication regulation? Not the chromosome. The plasmid replication regulation genes are on the plasmids themselves, which means that plasmids self-regulate their population. How is this possible? If there is a mutant that does not regulate, the mutant will throw away all self-regulating ones, and still regulation exists. Okay, an extremely complex problem. People have looked at this problem from a molecular point of view, but this is a population problem. This is a behavior problem. This is an ecology problem. Okay, so this is about that. Now I'm going to take a few minutes more on one specific story. Okay, and that is the story of cheating, but this is cheating in the flower pollinator game. Okay. Now, pollination is something that everyone knows right from school, that flowers have nectar and the pollinators come and take that nectar, but in that act, they pollinate flowers, they cross-pollinate. Okay, so it is supposed to be a mutualistic relationship. It's not that straightforward. So one student who was a bad student, who failed in first year for 2 consecutive years or 3 consecutive years, and he could have been thrown out of the system. When I intervened, he was from a different college, not my college. When I intervened, I met the principal and said that he is a very, very talented student, and the reason that he is not able to pass is that he is talented. Okay, because talented students often find the curriculum extremely boring. Okay, so this was a student who asked me this question: what if a flower decides not to make nectar? Will it get pollinated? This is a crazy question, but it's an extremely important question. And then most of the pollinators have no way to know whether there is nectar or not before entering the flower. And if they enter the flower, the job is done. Pollination will happen anyway. So I will get pollinated if I don't make nectar because others are making nectar. That is sufficient. Okay, so if there is a mutant that does not make nectar, the mutant will still do as well, in fact better, because it is investing less, and with less investment it is getting the same reward. Okay. We pursued this question, and I'm going to try to make the story short. What came out of this question? Okay. So potentially pollinators can be, flowers can be cheaters, and pollinators can do nothing about it, except learning that this fellow is a cheater and not going to that fellow again. This is the only thing pollinators can do, but they might have pollinated before realizing this. Okay. So first we said that if this logic is true, then you should get empty flowers. You should get flowers that don't make nectar. Right? Now how do we go around? So we should, okay, I immediately remembered what we used to do as children. We used to go around and pluck flowers and taste the nectar. And I very clearly remember that not every flower tastes sweet. So the moment the guy said, I realized that it is very likely that not all flowers make nectar. Some are cheaters. Okay. But tasting this is a subjective thing, which science will not agree. Science will not, it needs objective. So how to test whether there is nectar or not? We had nothing in hand at that time. So someone suggested use the diabetes urine sugar strips. Okay, if that can detect sugar in urine, why can't it detect sugar in nectar? Okay, so we started using that. At a later stage, we have micropipettes and micro estimations of sugars and so on. But we started with a simple urine stick. And it turned out that when we started sampling different species, almost every species has some fraction of nectarless flowers. But the proportion of nectarless flowers differs between species. Right? Then we started wondering, why is it that in some species there are only 2% cheaters, and in some species there are 60% cheaters? Why so much of a difference? Right? Then we started realizing that what could be the nature's within code, because there is nothing called nature. Okay, it's all. There is behavior of a species, there is a behavior of a genome, but there is nothing like behavior of nature. Everything put together becomes nature. So nature doesn't have a purpose, nature doesn't have a central regulation or control system or anything. So it turned out that we were not the first one to think of this. Right? So there was a person called Graham Bell, a very famous person who was the curator of Smithsonian Museum for quite some time. He had observed cheating among flowers, but he had a very different explanation. Our explanation was different than his. So then how do we differentiate? How do we say whether our interpretation is better or his interpretation is better? By the way, there were half a dozen other interpretations. Then we made this into a model prediction matrix. It turned out that this way of analysis was new in literature. Okay, so when we published this, we got a comment from the reviewers that it is an interesting way of analyzing. I was surprised. To us, it was common sense. Okay, but in literature, it became a new method of analysis. Fine, so let me avoid some details. And then we came to mathematics in order to optimize what is the optimum proportion of nectars a plant should put in flowers, or what is the optimum amount of nectar. Okay, now optimization is a mathematical problem. So it came to mathematics, starting with pollination biology. Right? So then we did some mathematics, we had some interpretations, some predictions of that maths, which we could test with field data. The plant managed to put nectar in some flowers and not in others. Now this is a developmental biology question. Okay, so you start with one question, you can cover every field of biology. Now, that question is not yet addressed. No developmental biologists have actually addressed this question of why different flowers can have different characters when the genome is the same. Okay. So, let me skip something. Now, there is other part of the story which has not yet been addressed. Okay, we haven't gone into that. And that is, if flowers are cheating, how should the pollinators react? What should be the pollinator's best strategy when some flowers have started cheating? Right? This question is yet not addressed. Okay? If someone is interested, we can of course go into this question. Okay. So, through this sort of a random system with no preset goals, okay, no agenda, no planning, I found as a teacher an extremely rewarding system. And the rewards were multiple. So intellectually, this was endlessly stimulating. Okay. Most of the things, most of my research findings have come through thinking with undergrads. Okay. Then, this is the best environment for perspective. Undergraduation is the best environment for perspective taking. What is happening in today's frontline research is there is over specialization, and people just don't talk to each other. So, interdisciplinary subjects, interdisciplinary questions are not even asked. Forget about addressing them, right? Whereas undergrads are being exposed to various fields at the same time. So, teachers and students really can ask questions which go across disciplines. And so, this is the best environment for perspective taking. Then, hunting for non-conventional talent. Most of the students who ended up publishing international papers or presented in international conferences or got some or the other forms of rewards were not academically the toppers. Okay. They were from anywhere in the academic hierarchy. Okay. Some of them were, I just gave an example, a fellow who had failed in the first year twice, and third times he was on the verge of failing. Today he is a scientist in NCBS in Bangalore. Okay, yes. And that can happen just because he did not find his education interesting. The moment he was allowed to ask his own question, the whole thing became interesting to him. And then, after having done a project, after having the sense of having discovered something, then he had no problem in clearing his BSc, his masters, and so on. Within 1 year after his masters, his book was published by the Indian Academy of Science. Okay. So, hunting for non-conventional talent, a Katta system is the best way to do. The best way of learning science, and even better clarity of thinking. And to me, who is not, maybe I can be called introvert. I don't completely agree with that classification. But if you want to call me, you might call me an introvert. I'm not very comfortable talking to people and getting acquainted and so on. But the richness of my social life came through Katta interaction. Okay, so that was my richest social life. So these are the benefits to a teacher that I enjoyed. And more than that, since I also entered research, I found that whenever research gets frustrating, and that happens rather too often, okay, research is very much frustrating. And when it becomes frustrating, teaching is the greatest relief. Okay, so every researcher should spend some time in teaching. And for a teacher, I think that it is just natural that you do research. You just can't do teaching without research, because the moment you ask questions, that is the beginning of research. So, I will end with this very famous poem of Robert Frost's. Two roads diverge in a wood, and I took the one less traveled by, and that has made all the difference. Thank you. Any questions are of course welcome if there is time, and if not, you can email me anytime.
Algorithms for musical creativity.
Oh, I can talk about that for hours. Because I come from a music family, Okay, so music was my original passion. And when I learned computer programming, and I started teaching because I realized that computational biology, this was in the 1990s. Computational biology, I realized that time is going to expand in a huge field. So I started teaching programming to biology first year and second year students. Okay. And then at the end of a couple of months of programming, see, this is a story when Excel wasn't there, or Excel was just on the horizon. We had a software called Lotus, okay, and things like that. Yeah, so everyone learned a little bit of programming and use of data, handling data and so on, and then they had to do a project of their own choice. Okay. So one girl who was also interested in music asked me whether I can do anything in music, and then we started talking about it. Okay. And then it turned out that music has so many similarities with DNA, because DNA is a linear array of 4 bases. Music is a linear array, but not 4, there are 12 notes. Okay. And then there are some differences, some similarities and so on. So we said that let us do whatever happens to DNA to music. Right. So in DNA there can be point mutations, there can be deletions, there can be insertions, there can be inversions, there can be crossovers. So we took the notation of a few popular songs and started doing all kinds of mutations on this. At the end of, at every step, it will play back the mutated music to you. And then you have to give a feedback. According to your feedback, it will either delete it, or mutate it further, or keep it in memory, or start doing it. So we are actually simulating the process of evolution. At the end of this, we did what is called a Turing test. Okay, Turing test is originally the test for artificial intelligence. And what we did was there were 20 computer-made compositions, 20 human compositions. Okay, but not the popular ones. Okay, new human compositions. And I am a composer. Okay, so they were my compositions. And of course, I am not popular, so nobody knew my compositions. Okay, so then their sequence was randomized, and people were asked to pick out the computer compositions versus human compositions, right? And they couldn't. Okay, so the computer-made compositions were as good as human compositions, and although we started with a few known tunes, they were unable to trace it back to the original tune. Just as we are unable to trace our ancestry back to bacteria like unicellular organisms. It has changed so much during evolution. So music can change in a similar algorithm, and you have an algorithm for creating new music, which is as simple as mutation and selection, only 2 steps. Nothing more needed. Okay, so the ongoing projects are often discussed to new students, right? Then they come forward and say that, oh, this is interesting, I want to work on. We never allot any project to anyone anytime. No, that is a sin. Don't give any project to anyone.